I want to start by setting a scene because I think it's a scene we all kind of romanticize, but in a way that almost misses the entire point.
Mm-hmm.
We have this picture of Gregor Mendel, right? This quiet monk just kind of pottering around a monastery garden in Brno.
Maybe humming a little.
Exactly. Humming a tune, picking some peas for dinner, and then whoops, he just stumbles upon the fundamental laws of genetics.
Right? A happy accident.
But when you actually dig into the sources, the transcripts of his lectures, the sheer amount of data he was working with, that whole image just, it completely falls apart. This wasn't a hobby.
Not at all. This was a high-stakes research facility that just happened to be disguised as a monastery.
A research facility, that's a great way to put it.
And you have to understand the context of Brno at that time in the mid-1800s. It wasn't some sleepy little town. It was the textile capital of the entire Austrian Empire.
Okay, so there's an industrial angle here.
A huge one. There was this intense economic pressure to understand breeding, not just for plants, but for sheep. They needed better wool, more consistent wool. Heredity wasn't an academic curiosity. Their economy depended on it.
So Mendel isn't just a monk with a green thumb. He's operating at this intersection of philosophy, physics, and what is essentially industrial research and development.
Precisely. And he was tackling a problem that had completely stumped the best minds in Europe. The main idea back then was something called blending inheritance.
The paint pot theory.
The paint pot theory, exactly. You take a tall parent and a short parent, and you get a medium child.
Like mixing red and white paint to get pink.
Yes. And the critical problem with that idea, the part that makes it a dead end, is that once you have pink, you can never get pure red or pure white back. The original variation is lost forever.
Which, if you're trying to figure out evolution, is a massive problem.
It's a disaster for Darwin, who was actually wrestling with this exact issue at the same time. If every new trait just gets blended away into some muddy average, natural selection has nothing to work with. Variation, the fuel of evolution, would just disappear.
So Mendel, with his background in physics and math from the University of Vienna, he looks at this blending idea and basically says,
the math doesn't add up. He suspected biology wasn't like mixing fluids. He suspected it was particulate, made of discrete, countable units.
And that is really the mission of this deep dive. We're going to unpack how Mendel proved that inheritance is particulate, that we're all built from these digital units, not analog fluids.
And he did it with the monohybrid cross.
It sounds like, intimidating jargon, but it's really just a beautifully simple idea. It's about isolating one variable at a time. It's the scientific method applied to heredity.
Isolating variables is the perfect phrase. That's really the key to the whole thing. And it's why he chose the garden pea, Pisum sativum.
This wasn't a culinary decision.
No. It was a rigorous engineering decision. To do proper statistics, you need massive amounts of data. You need what we now call big data. If he'd use, say, sheep, he'd get 1, maybe 2 lambs a year.
Not a great sample size.
Terrible. But with peas, he could get thousands of data points, thousands of offspring in a single growing season.
And he had total control over who the parents were. That's the other part of this that stood out to me from the sources, the idea of true breeding lines.
Right. Before he even started his main experiments, he spent 2 full years just calibrating his equipment, so to speak.
What does that mean, to have a true breeding line?
It means that if the plant is allowed to self-fertilize, its offspring are always identical to the parent. A true breeding tall plant only ever produces more tall plants, generation after generation.
No surprises.
0 surprises. Genetically, we now call this being homozygous, having 2 identical copies of a gene. Mendel had to be absolutely certain that his starting players, his parental or P generation, were genetically stable constants. He couldn't have any noise in the signal before he even began the experiment.
Okay, so he's got his pure lines. His tall plants are definitely tall. His dwarf plants are definitely dwarf. But peas naturally self-pollinate. That's great for keeping a line pure, but
it's terrible if you want to force them to cross with a different plant.
So he had to intervene. He had to perform this procedure called emasculation.
It's basically microsurgery on a flower. You have to very carefully open the flower bud before it's fully mature and physically remove the anthers.
The male parts that produce the pollen.
Exactly. And if you miss even 1 grain of that flower's own pollen, it can fertilize itself, and that entire data point is contaminated. Your experiment is ruined.
The level of patience and tediousness is it's mind-boggling. He would have had to do this for thousands and thousands of flowers.
And then, after emasculating the mother plant, he'd have to go to his chosen father plant, collect its pollen on a tiny brush, and manually dust it onto the stigma of the mother.
He wasn't just observing nature. He was forcing it to answer a very specific question.
That's it. And the question was this. If I cross a pure-breeding tall plant with a pure-breeding dwarf plant, what happens to the offspring? That is the monohybrid cross, tracking just one single character.
And he also did something called a reciprocal cross, which I found really insightful because it directly challenged some of the gender biases of the time.
Oh, absolutely. The prevailing thought was often that the male provided the spark or the essence of the trait, and the female was just the incubator.
The soil for the seed.
Pretty much. So Mendel tested that. He did the cross both ways. He put tall pollen on a dwarf stigma, and then he did the reciprocal. He put dwarf pollen on a tall stigma.
Yeah, what did he find?
The results were identical. It didn't matter which parent provided the pollen. For these particular traits, the genetic contribution was completely symmetrical. A huge insight.
So let's get to the data. He makes this cross, pure tall versus pure dwarf. The old blending theory says you should get
A field of medium-height plants.
But that is not what he saw. The F1, or first filial generation, they were all tall.
100% tall. Every single one.
So this is the first major shock to the system. The dwarf trait didn't just blend in and make the plants a little shorter.
It completely disappeared.
Yeah.
Vanished. It was as if the dwarf parent had never even been part of the cross.
That result, on its own, basically kills the blending theory.
It's a fatal blow. But it opens up a brand new mystery. Where did the dwarf trait go? Was it destroyed? Was it erased?
And this is where Mendel's patience really pays off. He doesn't stop with the F1 generation. He takes those F1 plants, the ones that look tall but had a dwarf parent, and he lets them do what peas do naturally.
He lets them self-fertilize to create an F2 or second filial generation.
And this is the moment. This is where the lost trait comes back.
The return of the repressed, you could say. In that F2 generation, suddenly he sees dwarf plants again.
And not just randomly, not just one or two here and there.
No, they appeared in a stunningly predictable and consistent ratio. When he did the experiment on stem height, his actual data, he counted 1,064 F2 plants.
Okay.
Of those, 787 were tall and 277 were dwarfs.
I actually did the math on that before we started. That comes out to a ratio of 2.84 to 1.
Which is, for any statistician, an incredibly clean 3-to-1 ratio.
And this wasn't a one-off. He found the same pattern for all 7 characters he studied.
That's the key. Whether it was round versus wrinkled seeds or yellow versus green cotyledons, when he performed the monohybrid cross, the F2 generation always showed this magical 3-to-1 ratio.
That consistency is what told him he wasn't looking at random noise. He was looking at a fundamental law of nature.
If it had been 3.1 for height, but say 5.1 for seed color and something else entirely for flower position, he would have been lost. But the universality of the ratio meant there had to be a model, a mechanism that could explain it.
Which brings us to Mendel's postulates. He basically had to invent a theoretical framework from scratch to explain the math. The first one is unit factors in pairs.
Right. He proposed that every character, height, color, whatever, is controlled by these discrete things he called unit factors. We now call them genes, or more specifically, alleles.
And he said they come in pairs.
Crucially, they come in pairs. In a diploid organism, like a pea plant or a human, you have 2 slots for the height gene. You get one factor from your mother and one from your father.
But in his F1 plants, those 2 slots were filled with different information. One factor for tall and one factor for dwarf.
JJ.
And yet the plant only looked tall. This leads to his second postulate, the concept of dominance and recessiveness. And I want to pause here because I think dominance is a word that gets really misunderstood. We imagine the dominant gene is stronger, or it's beating up the recessive gene.
Yeah, that's a very common misconception. It's not a fight. It's almost always a matter of function versus non-function.
What do you mean by that?
Okay, so think of a gene as a blueprint for making a functional protein, like an enzyme. The tall allele, let's call it T, is a working blueprint. It tells the cell how to make an enzyme involved in producing growth hormone.
So the plant grows tall.
Right. Now, the dwarf allele T is usually just a broken version of that blueprint. It has a mutation, so it either produces a non-functional enzyme or it produces no enzyme at all.
So it's not actively making the plant short, it's just failing to make it tall.
Precisely. So now think about the F1 hybrid. Its genotype is T. It has one working blueprint and one broken blueprint.
But one working blueprint is enough.
It's enough. The cell can still make enough of the growth enzyme to allow the plant to reach its full height. So the presence of the functional allele masks the effect of the non-functional one. That's dominance. It's function masking non-function.
Which also explains why the only way to be a dwarf plant is to have 2 copies of the broken blueprint.
Exactly. You have to be TT. Only then do you have a complete absence of the functional enzyme.
Okay, that clarifies so much. So the F1 plants were all tall because they were carriers of the dwarf blueprint, but the working copy covered for it. But the real genius, the absolute core of Mendel's discovery, is in his third postulate, the law of segregation.
This is the big one. Some call it the purity of gametes. Mendel had to explain how the dwarf trait could hide in one generation and then reappear, completely unchanged, in the next.
Right. It wasn't diluted or blended.
No. He proposed that even though those 2 factors for tall and dwarf coexist in the body of the F1 plant, they don't contaminate each other. They stay separate. And when that plant goes to make its own reproductive cells, its gametes, the pollen and ovules, those paired factors physically separate from each other,
They segregate.
they divorce. Each gamete receives only one of the 2 factors, and it's a 50-50 random chance which one it gets.
So the dwarf T allele, which has been hiding out next to the big T allele, separates out cleanly. It's still a pure T.
It's completely pure. It hasn't been changed at all. And that physical act of separation, of segregation, is the probability engine that creates the 3-to-1 ratio in the next generation.
It's amazing. He figured all this out just from the numbers. I mean, we need to bridge the gap here between Mendel's abstract math and a physical reality inside a cell.
Yes, because he was working in a total black box. He inferred that these factors had to exist because the math demanded it, but he never saw one.
Yeah.
He couldn't point to a gene or a chromosome.
That discovery came much later.
Decades later. It wasn't until around 1902-1903, with the work of Walter Sutton and Theodore Boveri, that we finally found the hardware that was running Mendel's software.
That's a great analogy.
They were cytologists, looking at cells dividing under the microscope. And they noticed these little thread-like things, chromosomes, that were behaving in a very peculiar and specific way.
A way that looked familiar.
A way that looked exactly like Mendel's factors were supposed to behave.
Specifically during the process of meiosis, right? The cell division that creates gametes.
Right. In a normal diploid cell, you have pairs of homologous chromosomes. One chromosome in each pair came from the mother, the other from the father.
Okay.
During a phase of meiosis called anaphase, these homologous pairs line up at the center of the cell, and then they are physically pulled apart to opposite poles of the cell.
That moment, that physical pulling apart, that is the law of segregation.
That is the physical manifestation of the law.
Mm-hmm.
If the gene for P height is located on, say, chromosome 4, then in the F1 hybrid, one copy of chromosome 4 carries the T allele, and its homologous partner carries the T allele. When those chromosomes were pulled apart in anaphase I, the alleles go with them. They end up in different daughter cells, which will become the gametes.
The biological machinery perfectly mirrors the statistical model he created. It's so satisfying when it all just clicks together like that.
It's one of the most beautiful syntheses in the history of science.
So speaking of models and rules, let's talk about the toolkit we use to predict these outcomes. The Punnett square.
Ah, yes.
We all did these in high school biology, but I feel like we often just fill in the boxes without thinking about what it actually represents.
It is, at its heart, a probability matrix. It's a visual tool, named after Reginald Punnett, that helps us map out the outcomes of random fertilization.
So how does it work, just to refresh?
You take the gametes produced by one parent and list them across the top. You take the gametes from the other parent and list them down the side. For our F1 self-cross, each parent produces 2 types of gametes in equal numbers, T and T.
So you have T and T on the top, T and T on the side, and the boxes are just the possible combinations.
The 4 possible ways that a sperm can meet an egg, and this is where we have to be really clear about the difference between genotype and phenotype.
Genotype is the genetic code, the letters. Phenotype is the physical trait you actually see.
Exactly. So if you fill in the Punnett square, you get a genotypic ratio. One box is TT homozygous dominant. 2 of the boxes are T heterozygous.
Right, you can get a big T from dad and a little T from mom, or a little T from dad and a big T from mom.
Correct. And the 4th box is T homozygous recessive. So the genotypic ratio is 1 TT to 2 T to 1 TT.
A 1.2.1 ratio, but that's not what Mendel saw in the garden. He saw 3.1.
Because of dominance. The TT plant and the 2T plants all have at least one functional T allele.
So they all look tall.
They all have the tall phenotype. So you collapse those categories. The 1 and the 2 add up to 3. And you see 3 tall plants for every 1 dwarf plant. The 3.1 phenotypic ratio is a direct consequence of the 1.2.1 genotypic ratio plus the principle of dominance.
One thing the lecture notes and the sources really hammered home is that these are probabilities, not guarantees.
No, absolutely not. It's just like flipping a coin. The probability of heads is 50%, but if you flip it 4 times, you could easily get 4 heads or 4 tails.
It's the law of large numbers.
Exactly. You only start to see the ratio reliably emerge over a large number of trials. That's why it was so important that Mendel counted thousands and thousands of peas. If he'd only grown a dozen plants, the random statistical noise might have completely obscured the pattern. He might have missed it entirely.
So this brings up a really practical problem for a breeder or for Mendel himself. You're standing in your garden, you see a tall plant. The problem is you don't know its genotype just by looking. It has a genetic poker face. It could be TT or it could be TT.
And you need to call its bluff. You need a way to figure out what genes it's hiding.
And for that, there's a brilliantly simple tool called the test cross.
It's one of the most elegant pieces of scientific logic. You have your mystery dominant plant, let's call it P question mark. To find out what that question mark is, you cross it with a very specific partner.
A tester.
A tester. And the tester is always, always the homozygous recessive individual.
So the dwarf plant, the TT, why that one specifically?
Because the homozygous recessive is genetically transparent. It's like a blank slate. We know with 100% certainty that it can only contribute recessive teeth heels to its offspring. It has no dominant teeth heel to mask or confuse the results.
It's the control group.
It's the perfect control. The phenotype of the offspring will therefore be determined entirely by what the mystery parent brings to the table.
Okay, so let's play detective. I take my mystery tall plant, I cross it with a dwarf plant, and then I look at the kids.
You plant the seeds and see what grows. There are only 2 possible outcomes. Scenario A. Your mystery plant was purebred tall, TT.
Okay, so it can only make T gametes.
Right, and the tester can only make T gametes.
Yeah.
So every single offspring will have the genotype TT.
Which means they will all be?
All tall, 100%. So if you look at the offspring and they're all tall, you can be very confident your mystery parent was TT.
Okay. But scenario B, what if my mystery parent was a hybrid TT?
Well, now it's making 2 kinds of gametes. Half get the T allele, half get the T allele. The tester is still only giving T.
So half the time you get TT.
And you get a tall plant.
And the other half the time you get TT.
A dwarf plant. So your offspring will be a mix. Roughly 50% tall and 50% dwarf, a 1-to-1 ratio.
So the second you see even one single dwarf plant pop up in that test batch.
Game over. You know for a fact that the tall parent must have been carrying that hidden T allele. The appearance of the recessive phenotype in the offspring unmasks the heterozygous genotype of the parent.
It's so simple and so powerful. Now, the sources also mention a back cross. I sometimes see that used interchangeably with test cross. Is it the same thing?
It's a subtle but important distinction. They're not exactly the same. A test cross is a type of back cross.
Okay, so all test crosses are back crosses, but not all back crosses are test crosses.
You got it. A back cross is just the general term for crossing any hybrid offspring, like from an F1 generation back to one of its original parental types.
Either the dominant parent or the recessive parent.
Right. And a test cross is the specific case where you cross it back to the homozygous recessive parent for the purpose of figuring out the genotype.
So what's the use case for the other kind of back cross? Why would you cross a hybrid back to its dominant parent?
That's used all the time in modern genetics, especially in things like creating mouse models for diseases or in what are called gene knockout experiments.
How does that work?
Let's say you've discovered a really interesting new mutation in a mouse, but that mouse's overall genetic background is, well, it's a mutt. It's messy. If you want to study just your one mutation, you need to isolate it in a clean, standardized genetic background.
So all your lab mice are identical except for that one gene.
Exactly. So you take your mutant mouse and cross it with a mouse from a pure inbred lab strain. Then you take their offspring and cross them back to another mouse from that pure strain. You keep doing that generation after generation, always crossing back to the pure strain parent.
You're essentially diluting out the original messy genome.
You're diluting it until after maybe 10 or 20 generations, you're left with a mouse that is 99.9% genetically identical to the pure lab strain, but it just happens to carry your one specific mutation of interest. It's a technique called introgression, and it's pure Mendelian logic.
Wow. It's just incredible that a method that was conceptually designed for 19th century pea breeding is still the standard operating procedure in 21st century genetic engineering labs.
It really speaks to the fundamental robustness of the system. Mendel didn't just uncover a rule for peas. He uncovered the basic logic of diploid heredity.
There's also a quick note in one of the sources about about notation. We've been using capital T and lowercase t, which feels pretty standard, but it mentions that if you go talk to, say, a fruit fly geneticist, they speak a different language.
Oh, yes. The Drosophila people. They have their own entire dialect. They work off the concept of the wild type.
The wild type. What's that?
The wild type is just whatever is considered the standard, normal, most common version of a trait found out there in nature. And they mark the allele for the wild type with a simple plus sign.
Okay, so no capital letters for dominant, just a plus I.
Right. And they named the gene after the first mutation that was discovered, not the normal version. So, for example, there's a mutation in fruit flies that gives them an ebony body color instead of the normal grayish-brown. The gene is called the ebony gene, symbolized E.
So the recessive mutant allele is just E.
And the dominant, normal, wild-type allele is E plus E. It can be really confusing if you're switching between textbooks, but the underlying logic of segregation, dominance, all of it, it's exactly the same.
It's just a different user interface on the same operating system.
Perfect analogy.
As we start to close the loop on this, I want to zoom out a bit. We've gone from the monastery garden to the chromosome to the Punnett square, but I keep coming back to this idea you mentioned at the start, the digital nature of this. Before Mendel, biology was seen as fundamentally analog. It was all blending fluids and smooth curves. Mendel is the person who turned biology into bits, into zeros and ones.
That is the profound philosophical shift. It's the quantization of biology. Just like Max Planck later discovered that energy doesn't come in a smooth flow, but in discrete packets called quanta, Mendel discovered that biological information comes in discrete packets called genes.
And this is so important for preserving variation.
It's everything. If inheritance really were an analog blending process, any beneficial new mutation would be like a single drop of black ink in a swimming pool. In a few generations, it's just diluted into nothingness. It's lost.
But with digital particulate inheritance, that drop of ink, that new allele, it stays a concentrated packet.
It can be passed down intact. It can be hidden for a generation in a heterozygote, then reappear in its full undiluted strength later on.
That's what allows evolution to actually work.
Yes, recessive alleles can hide out in the population, sheltered from natural selection in carriers, creating this vast reservoir of hidden genetic diversity. And that diversity can be unleashed when environmental conditions change. Without Mendel's discrete particles, natural selection quickly runs out of fuel.
So Mendel didn't just explain why you might have your grandmother's blue eyes. He actually provided the missing mechanism that Darwin's theory of evolution desperately needed.
He laid the mathematical foundation for all of modern genetics.
So one final thought for you to chew on as a listener. We give Mendel, and rightly so, a huge amount of credit for his scientific rigor, his patience, his mathematical mind, but we should probably also give him some credit for his luck.
Oh, a massive amount of luck. An almost unbelievable amount.
Because the 7 traits he chose to study in the pea plant, they all just happened to be on different chromosomes, or at least they were far enough apart that they behaved as if they were. They all assorted independently.
And just as importantly, they all showed this very simple, complete dominance. One allele completely masked the other.
Right. If he had happened to pick a trait that showed incomplete dominance, like in snapdragons, where crossing a red and a white flower really does give you a pink flower.
He might have looked at his F1 generation and concluded that the blending theory was right all along. He would have confirmed the old dogma instead of overthrowing it.
Or if he'd picked 2 traits that were linked, stuck really close together on the same chromosome, his neat and tidy ratios would have all gone messy. He would have never seen that clean 3-to-1 signal through the noise.
He could have spent his entire life staring at confusing data, concluding that biology was just too chaotic to have simple laws.
It's a real lesson for experience, isn't it? Sometimes a breakthrough isn't just about asking the right question. It's about having the luck to ask that question to the right organism.
The pea plant was the perfect accomplice. It simplified reality just enough for the human mind to grasp the beautiful, simple laws hidden underneath. We had to find the simple threads first before we could ever hope to untangle the complexity.
It makes you wonder what other fundamental laws are just hiding out there in plain sight, waiting for someone to pick the right vegetable.
That's the exciting part. We're still looking.
Thanks for joining us on this deep dive into the roots of genetics. It's a story of patience, probability, and the simple power of counting your peas. We'll catch you on the next one.